I'm going to go ahead and I am recording this and I'll be putting this on Facebook Live. So just so you are aware of that, and it will be available later on YouTube, probably tomorrow or so when I get that uploaded. So we are continuing to let some folks in as they stream in. So, but I'll go ahead and get started because I think we have a pretty full slate of folks to talk about some cool research and projects here. So we want to get started. So thanks everybody for attending the Engineering in Action. So I'm gonna flip this over to, we have a student host, Mariah, who will introduce, I believe, or do I introduce Dean? I think you talk first, Christo. She'll say a few words to kind of kick us off here and get us started, and then Mariah will be the host. So she will be taking questions in the chat later. So anytime you know, especially as they end, if you have questions, just feel free to chat them and she'll be moderating those. So we should be ending about 4:30. So they'll be giving a presentation, and just make note of any questions or points you want to elaborate on. So I'll go ahead and turn it over to you, Christos, to say a few words.
Yes, thank you, Kim. I would like to welcome everyone to this very modern episode of Engineering in Action. I mean, we all know COVID has been a phenomenal issue for the entire universe, not just one country. So I'd like to thank the speakers to be, because it does show how good we are in the area of engineering. And with that, I will thank you ahead of time, and I'm gonna, you know, listen and learn. So thank you. Mariah, you're next.
Thank you, Dean Christodoulou. Good afternoon, everybody. I hope you're having a wonderful Friday. My name is Mariah Paiuchuli. I'm a senior studying chemical engineering. It is my pleasure to be the host for today's Engineering in Action speaker series, A Bright Future, the virus-killing possibilities of UV light in polymers. So today our talk will be presented by Drs. David Whitten, Eva Chi, Linnea Ista, and Alison Kell. The research you will hear about has transpired from a cross-campus collaboration between the School of Engineering and the School of Medicine. The team is combating the pandemic and future threats by using UV light and polymers. Before we get started, I would like to mention that we do have a time reserved at the end for questions and answers. So as always, we encourage you to use the chat function located at the bottom of your screen for any of your questions. And it is now my pleasure to introduce Dr. David Whitten.
Thank you. I'm very happy to have a chance to tell you about what we've been doing, about a very nice collaborative research project that got started just about a little less than a year ago. So may I have the first slide? I think you.
Yes, I'm sharing the, sharing the screen, right?
No.
Oh, not yet. Okay.
I thought you were just going to show the slides.
Yes. Mm-hmm.
Okay.
Okay, there you go.
Okay. So basically what we've been looking at is shown here in the title, the virus killing possibilities of ultraviolet light and polymers. And I should also say oligomers, small version of polymers. And this has been a collaboration between primarily four of us, Eva, Linnea, Alison from North Campus, and myself in the Center of Biomedical Engineering and the Department of Chemical and Biological Engineering at UNM. Okay, on the next slide, this is just a schematic. We have these materials that we've synthesized, and they often absorb visible light, such as this vial shows with a sample that absorbs and is red. They're highly fluorescent, but most importantly, under UV or visible light, they produce very quickly several log kills or inactivation of the SARS coronavirus 2 in 10 minutes, actually much less than 10 minutes in certain cases. So if we go to the next slide, Yeah, so this started a little over a year ago, and we were responding to the governor's call, state of public health emergency, and invoking the powers. So let's go on to the next slide.
So I want to add that, we all remember last year this time that all of us, have, since the first few cases of COVID-19 was diagnosed, we all had to make a huge transition. At the time, all of us had to scramble with the lab being shutting down and transition to teaching online, as well as dealing with kids at home, et cetera. So, and amidst all of that we had to deal with, one question that many of us had, is that what can we do to contribute towards combating the pandemic? Many of our colleagues jumped right into action. This is my colleague, Heather Canavan. She started making masks and 3D printing pieces, that go into the mask in her garage. She mobilized friends and family, neighbors, and students. And another example is Professor Christina Salas, who's given a previous engineering in action talk. She started making 3D printing much needed personal protective equipment that was in short supply earlier in the pandemic. So the question that we had for us, and the solution, or the solution that we came up with, and perhaps it's obvious in hindsight, is that we have been working with a class of synthetic compounds, small ones being oligomers, and larger compounds being polymers. This was really the work that Dave Whitten started about 15 years ago, and I started collaborating with him about 10 years ago, trying to develop these compounds and also understanding how they work. The bottom line is that these compounds, some of these examples are shown here, based on this molecular architecture, show remarkable biocidal activities. That is, they're highly efficient at killing a broad range of microbes, including bacteria. This is one example of E. coli before and after, yeast, spores, fungi. And we also remembered back in 2011, we published a paper on how these compounds are also active against bacteriophages. These are viruses that can kill bacteria, that infect bacteria. It's not really a big concern for human health, but nonetheless, we start to think, wait, might these compounds also be active against the SARS-CoV-2 virus, which causes COVID-19? So following our conversation, we talked to our colleague and good friend, Linnea, and see how we might be able to test our compounds.
Yeah, because as you know, this is a dangerous compound. And so we did not have the facilities in the Center for Biomedical Engineering to do this. And I am fortunate enough to be on the campus biosafety committee. And I knew a couple of my colleagues there were working on COVID. And so I contacted them, and they introduced us to Dr. Alison Kell, who kind of completed the team. So Eva and Dave and Kirk and I had been working on some of the bacterial stuff for years, but we really needed that part with Alison, and she jumped right in and came up with a very, very cool, simple assay that allowed us to test these. And as soon as we got back from COVID shutting down the lab, we started this, I believe, in June. And by December, last year, we produced a paper showing that we could indeed kill SARS-CoV-2 by using these compounds.
So Dave is going to tell us about the compounds that we initially selected to test.
Yes, so we decided we couldn't test all of the compounds we had made and studied, but we felt that we'd take representative materials from our group. And we looked at, first of all, the poly 4 and poly 5 were the two polymer systems we looked at extensively with bacteria and earlier viruses. They are positively charged. They're water soluble, but they very much are attracted to pathogens which have an overall negative charge. We've also made the oligomers 1, 2, and 3, and we decided to check this panel. And so you can see them, they're different colors. Some of them absorb in the visible, some of them absorb more strongly in the near ultraviolet. But we made up solutions. These are solutions Eva and I prepared in the lab. And then we sent them to Alison, and Alison took it from there.
Yeah, so this will be the virology section of the talk. I think everybody's pretty familiar with the SARS coronavirus 2 at this point. So I'll keep this little background brief, but just as we know, the SARS coronavirus is a single-stranded positive sense RNA virus. So the nucleic acid is shown here. And it's surrounded by a protein coat, which is then surrounded by a lipid membrane. The lipid membrane is then studded with these spike proteins. And it's these spike proteins that interact with the host cell, specifically ACE2 or TMPRSS2, that allow it to gain entry into the cell. And then once it gains entry into the cell, the fusion of that membrane surrounding the viral particle with the host cell, the somal membrane, is what allows the release of the nucleic acid into the cell and to establish that infection. And so we were wondering if some of these polymers that had been previously developed and tested against other bacteria or other viruses might also interact with the viral proteins or the lipid membrane in order to have a similar effect in reducing infectivity of the virus. So just kind of before we get into the data, I wanted to get us all on the same page as far as the techniques that we use in the lab in order to visualize infectious particles. As we know, this virus is spread through the air. We can't really see it when we're spreading it around, but we can, but what we see in the laboratory.
Sorry, guys.
Oh, that's okay.
We saw the animation again.
So we can't actually see it with our bare eyes, nor can we really see it kind of under a simple microscope. But instead, what we can do is visualize infection. And so what we do is we take a Petri dish that has a complete monolayer of cells that are susceptible to viral infection. We incubate those cells with media that contains infectious virus. And as you can see here, the schematic is really showing that that media contained about 6 viral particles that were able to establish infections in 6 individual cells all across the plate. Now, the way that we set up these assays, once we allow the virus to kind of incubate and sit within that monolayer, and the virus is able to replicate and spread to neighboring cells. We set up this assay so that they can only spread to those neighboring cells, and they create kind of these patches of infection across that cell monolayer. But then what happens is that these cells die following SARS coronavirus infection. And so those dead cells leave empty spaces all over the tissue culture dish. And then once we counterstain for live cells, that counterstain can reveal holes in that tissue culture dish that represent the individual infectious viruses that were able to establish the infection. And so here's a nice picture of a plaque assay, which we have this purple counterstain. So any purple across the cell monolayer is in like intact cells. So those are live cells, and all of those little white dots are spaces that are dead cells. So those are places in which a viral particle landed, started an infection, and then created those dead cell spaces. And so we can count all of those white dots, and that allows us to back calculate how many infectious particles or plaque forming units were actually present in the initial inoculum. And so using this assay, we were able to ask if we pre-incubate a viral containing media with some of these compounds, can we inhibit plaque formation or infection in that cell monolayer? And so what we do, shown here in the schematic, we start with media containing virus and also individual compounds. We incubate the virus plus the compound in this photo chamber, which is shown here in this picture, which is really just a chamber that contains a bunch of bulbs that can emit light at different wavelengths. And then after incubating within this light photo chamber for a given period of time, we then treat the cells in a monolayer and count the plaque forming units that are present within that solution following incubation with our compounds. And so, so now I'll just jump into the data. So what we found, looking just specifically here at the oligomers that we tested first, what we have starting with, oligomer 1, on the y-axis is log plaque forming units per mil. So that's really the infectious titer of the solution following incubation with oligomer 1. And what we see is that as early as 10 minutes following incubation of oligomer 1 in UV light, we see about 99% killing. So an incredible reduction in log infectious units within that solution. And at 15 minutes, we're almost to complete killing or virus inactivation. And by 20 minutes, we don't detect anything at all. Similarly, for oligomer 3, we see very quick inactivation of the virus following incubation with that compound. And that was even on the order of 5 minutes. So already dramatically reducing the viral infectivity in that solution. Oligomer 2, we didn't see as dramatic an effect, but we still saw a statistically significant reduction over time with significant inactivation of the virus following incubation in the UV light. Importantly, so panel D, what that's showing is that even the UV light itself, we know does inactivate the SARS coronavirus and many other viruses. And so if we incubate the virus media only without any oligomer whatsoever in the UV light in that photo chamber, we do see reduction, but we don't see it on the as quickly without the oligomers just in the UV light. We do see reduction by 60 minutes, but we don't see much before that. And so that indicates that these compounds really do facilitate the inactivation of the virus in UV light. And just on the far panel is a little plaque assay just to show you what we're actually counting in the lab. So then moving on to the polymers that we tested. Importantly, what makes this panel a little bit different is that we tested the polymers both in 10 micrograms per mil, but also in 5 micrograms per mil, trying to identify how little of this compound is really required to inactivate the virus, either at in a light chamber that's emitting a range of 300 to 400 nanometers, or lights that are emitting just at that 420 nanometer wavelength. But what we found that polymer 4, while it wasn't quite as effective at inactivating the virus at the early time points, eventually we did see a significant reduction of viral infectivity following incubation in both light contexts. Polymer 5 was actually very efficient as early as 20 minutes. I mean, pretty good as early as 10 minutes, I'll say too, but very nicely reducing it down to that 99.9% infectivity at 20 minutes post-incubation. And we could even go down to that 5 micrograms per mil concentration. We don't really see a significant difference between its ability to inactivate the virus, suggesting that it's possible that we could even go lower in concentrations and see a real effect in the inactivation of this virus with these polymers. So then, the real test that we also wanted to ask, right? So we could incubate virus in UV. We know that UV kills virus. But we don't all just kind of walk around in UV chambers all day. So how can we really make this applicable for the real world? And so we wanted to test some of these compounds just in fluorescent light exposure. So this is going to mimic, hanging out in your cubicle all day exposed to fluorescent light bulbs. Can we apply these compounds in a way that would make them active against SARS coronavirus just in the fluorescent light conditions? And what we find is that 2 polymers that we tested, and I should mention this wasn't published in the previous paper, so this is exciting data that people haven't actually seen, is that 2 of the polymers that we've looked at actually do have significant inactivating properties as early as 30 minutes post incubation. So very quickly able to inactivate these viruses just in the standard fluorescent light conditions. And as you can see on the far end of the panel, just with water, incubating virus with water and incubating them in these light chambers, you really don't see a significant reduction in infectivity, showing that these compounds are really doing an impressive job at inactivating the virus just in standard light conditions, which is really exciting. So what makes this so exciting? I think one of the things is that, having a potentially broad acting compound, something that can be effective against multiple viruses, not just kind of the pandemic of the day, as we like to talk about it, that something that might be able of combating something that we have as existing, like flu virus that circulates every year, or other emerging viral infections, such as Ebola virus, which is coming back in Africa now, and also hantaviruses, which are of particular interest here in New Mexico. If we have something that could act against all of those, that's much preferred than kind of developing de novo new compounds and new drugs for every single viral threat that we come across. Interestingly, or I guess what makes us really excited as researchers is that we don't yet know the specific mechanism of action. And so if we can identify potential mechanisms of action, we might also be able to have some more rational design of some of these compounds and make them even more effective going forward. And importantly, there's this quote here from our famous Dr. Anthony Fauci, who wrote during the Zika virus outbreak that, we don't need just a one bug, one drug approach. What we really need are these broad spectrum, either antiviral or some sort of disinfectant or preventative measure that can act against all classes of viruses.
So why we're excited about these compounds is the applications. We started thinking about this like almost as soon as, oh my God, what are we going to do with these? As soon as Alison gave us our first set of data saying they work. So that the biggest thing we thought of right away is masks, right? If you have these on the outside surface of a mask, right? And you walk, and now that we know that they're exposed, that we can kill the virus with light, you could wear a mask and be simultaneously protecting yourself and killing virus as it comes in. So it's more efficient masks. I have a feeling that they're going to be even more efficient if you go out in the bright sunlight, right? Other approaches that we're thinking about are longer lasting wipes. So you could have these on a wipe and leave the wipe out and kill the virus that way, take it off the surface. And another one, actually this was Eva's idea this morning, so I was pretty pleased with this, is ventilation and filter systems. So if you have these on the outside of a filter in the presence of light, as the virus comes through, like in the airplane, I'm traveling in very soon, and thinking about the circulation of this virus through the airplane, if the filter is exposed to room light or even a certain type of, or cabin light or a certain type of specialty light, that will help with the filtration. And currently we are in planning stages with some other colleagues in the School of Engineering to incorporate these compounds into silk. We also know how to attach them to other kinds of surfaces as well. And these are potentially superior to those that are currently under consideration by the FDA and EPA, because they are reactive under light that we see every day. A lot of the compounds that people are exploring, such as quats, are not that active against against this particular virus. And it's kind of continuous disinfection if you think about it. If you just leave it out, if these are attached and you leave it out under room light, right? You're continually, if you have this on your desk is continually being sanitized. And you don't necessarily have to come in every 2 hours and wipe it down.
So I think, again, I'd like to end by highlighting the fact that our materials are active under both ultraviolet and visible light. Visible light, like I have here in my office. And that's really something no other disinfectants that we know of right now have. And so if we go back to the last slide again, we'd be very happy to take questions you might have. Yeah, one more. Yeah, so please, any questions we'll welcome and try to answer them. Thank you very much.
Wow, thank you all. I think this is just amazing. Like, you know, you're our professors and you're doing something so incredible. So thank you all for sharing it with us and, you know, giving us all more glimpses of hope in this pandemic. So we'll start the question and answer portion now. Remember, you can use the chat function located at the bottom. It's like a little comment symbol. So our first question we have from Karen. She asks if they could be applied as a cream.
I would think so. We've done skin testing with our compounds and found that they are not corrosive on human skin models. And so I think applying them to and using them as a cream for skin and protection of that sort, that's just one possible application, but that's a good one.
Yeah. Our next question is from Juan. He asks, will these compounds be as reactive under LED lighting as compared to fluorescent lighting?
I think they would be comparable, show comparable reactivity. It's really the photon flux that is going to determine what the photon flux and the absorption of the material that really determines how efficient the process will be. And I think the efficiency is pretty good, so it should be fine under LED or fluorescent lights.
Okay, thank you. Gabriel asks, or they say, this sounds really neat. While these compounds are so good at killing viruses, do they have adverse effect on the organisms we're trying to protect?
Oh, go ahead, Eva.
I could tell you about, so one of the things that we have figured out, we still need to test these compounds, but the work that we have been doing in the last 10, 15 years also tells us that these compounds are selective. At the concentrations, the ones that Dr. Kell talked about, you know, the low micromolar concentrations, microgram concentrations, they can effectively kill bacteria, viruses, but they're not harmful to many different kinds of mammalian cells that we've tested. So in terms of these compounds are not just very, very active, but they're also selective as well.
And if we have them attached to surfaces and not like going into the waste stream, I think that you know, I'm a biofilms person, right? So that is a concern, right? We don't want to kill harmful or helpful organisms. I think if they're attached to a surface, it's not going to be, you know, kind of bacteria-geddon or anything like that.
Okay, thank you. Karen asks, would there be any major adverse reactions if someone uses it in a tanning booth?
I'm sorry, could you repeat the last part of that?
Yes. Would there be any major adverse reactions if someone used it in a tanning booth?
I don't think so.
I think there might be, right? Yeah.
Well, you're gonna get really dark and really clean.
Yeah, it would be very clean, but you know, putting them on your skin in a tanning booth would be bad. If you're putting it on the surfaces of the tanning booth, as Eva said, it would be super. That would be like the safest place in the gym, right? Is that particular place or that particular tanning booth.
Right. Dean Christodilo asks, what part of this work can we start using to sanitize our classrooms for the fall?
Where's the EPA approval, Dave?
I will pay, guys. I will pay. We need that. So
Okay. Well, this is a big problem. I mean, EPA registration is slow and expensive. EPA does have an accelerated process for materials like this. And we've already shown in peer-reviewed research that these things are very, very effective towards inactivating the virus. So I'm hoping we can get some accelerated approval through EPA. As soon as we get that, then we're ready to launch products. Certainly sprays and wipes will be some of the first products that can be used and be quite versatile.
Yeah, I was thinking between classes, you sanitize, you let the next group come in and do it all over campus. You guys will become millionaires.
Yeah, you wouldn't have
One
to.
semester.
I remember watching the inauguration and seeing that for every speaker that came up there beforehand, there was someone going around with a cloth and wiping it off, and that wouldn't be necessary with our materials.
Yeah,
that's what I think is most one of the biggest advantages.
So I think Alison could probably answer this question. What actually kills the virus, UV light or polymers?
Well, so we know that UV light can inactivate the virus. What we showed though is that the polymer plus the UV light really enhanced that inactivation, and it reduced the inactivation time on the order, of it was 5 minutes for some of those polymers or oligomers, and up to maybe 60 minutes. We do see inactivation of the virus just by UV light alone. So it's probably kind of a combination maybe, or, the polymer obviously needs some level of UV light in order to become active itself. But it definitely enhances it in the presence of UV light. It's not just the UV that's killing it.
Yeah, I think we know primarily what the mechanism is, and that is that the polymer absorbs light. The polymer or oligomer absorbs light, and then it goes from its first excited state, which is a singlet, and the state responsible for fluorescence, to a triplet state, which is very active at activating molecular oxygen. And so the major product that does the damage under light absorption is probably singlet oxygen or peroxy radicals that are generated subsequently by various electron and proton transfer processes. So we have a good idea of what this has caused. And the reason why, for example, that our materials are not active against the coronavirus in the dark is because the coronavirus is an enveloped virus, which has a protein coat. The protein coat attracts our materials. Some of it has positive charge, some of it negative charge. Our materials will dock against the protein coat, but they won't disrupt the protein coat. On the other hand, once you make singlet oxygen, the oxygen is very small and can easily penetrate the protein coat and even damage it. But certainly it's very reactive towards the nucleic acids in the cell.
Kim asks, where are you with the commercialization of this discovery? Will this be something that the layperson can utilize soon?
I would think so. Again, it's all the requirement for EPA registration, which slows down the process. We've done the tests which indicate that our materials are not very toxic towards mammalian cells, as Eva mentioned. And so I think there should be no barrier, but we do have a lot of bureaucratic hurdles to go through.
And we have filed a patent, so and there is a company developing these, so
that's awesome. Dave says, I have a Homedics UV air purifier. Can the polymer be placed on a filter inside a similar purifier to possibly eliminate coronavirus or other viruses?
Yeah, I think air filtration is a great application.
And if you've already got UV in there, right, if you just put that on the surface where it's exposed to UV, I think that'll work.
Yeah.
Okay. Karen asks, would a black light work? Like if you were to wipe down a classroom in between classes, then run a black light wand over it? Or would you change part of the light bulbs in the room to the UV lights, flip a switch, then inactivate the virus in between?
I think both of those are very good applications. Certainly UV light that costs very little could be sprayed over the area you're trying to decontaminate, disinfect.
Or if you attach them to the surface of the desks itself, right? If it was a coating that you attach to the surface of the desk, right? Then the room light should kind of provide this continuous deactivation of any virus.
Absolutely.
Okay, let's see. Okay. Kim asks, what is next for your research in this area?
What are the next?
Well, we've submitted a grant and partly to look at the question of the mechanism of action, right? And also our collaboration with, potential collaboration with people who are going to help us get it into fabric really quickly and efficiently.
So the next steps are both kind of looking more fundamentally at how these compounds work, which part of the virus do these compounds interact with, and how the activation actually occur, and to the other end of the spectrum, which is getting through EPA registration, and also working on some of these application areas that Linnea talked about. So we're working on all fronts.
Okay. Andre asks, can just a very powerful UV source sanitize a classroom?
It can, but it would be much more harmful to anybody around it. There are two kinds of UV. I mean, there's near ultraviolet, which is not very harmful, but still you don't want to be in the presence of it or look at it. And then there's deep UV, which is very reactive, but that's really very, very, it's a very dangerous source of energy.
Okay, thank
Yeah,
you.
and I think the other part of this is what Eva's trying to show us is that UV light does in fact inactivate the virus, but what's added by the incubation or the interaction between the compound and the virus is the rapidity at which this inactivation occurs is much greater with these oligomers and these polymers. So yeah, you could try to hang out for long enough, maybe 2 or 3 hours with a UV wand over the same spot and try to inactivate all of the virus that might be present. But it would be much more efficient to use these oligomers and polymers. And then especially if we're able to identify ones that are active in just the regular fluorescent light or sunlight, then that even negates the use of the UV at all. Because as Dave mentioned, yeah, it's very harmful to us, the UV light.
And it's harmful to materials as well. So you don't want to be zapping plastic with a lot of heavy duty UV light.
Right. So in this control experiment, it took 60 minutes of irradiation. And if you remember this picture, it's pretty intense UV. There are 10 bulbs, kind of irradiating these samples. So I would say, like what Alison said, yeah, unless you want to irradiate the same spot for 2 or 3 hours, just having the compounds there would definitely is much more effective.
Michaela asks, so is it going to be more challenging for you guys to eliminate it in more confined spaces, or places like airplanes?
No, I think it would be, I think having the air filtration was discussed a little bit, that's a really good application for them. The filtration and deactivation of the virus would really reduce the population very quickly. So that's a very ideal place where you have the constant filtration of the air. You'd constantly be removing the virus particles and destroying them.
Karen asks, could this be made in a spray form for easy application?
I think a spray form is really a great application and one which we are certainly exploring. The only problem with the spray is I don't think we'd be concerned that I wouldn't want to spray this into somebody's eyes. I think that would be, a problem.
Mhm.
And so I think we're thinking more in terms of sprays, that these sprays would be for industrial use, not for individuals using them where they have a chance of injuring themselves through the spray.
Okay. See, John asks, has there been commercial interest to take these ideas to market?
Yes, in fact, we have a startup company that we started about 3 or 4 years ago, and we're actively working with the former director of the EPA to get through the registration process. And I think we should be able to get accelerated review and approval, and then we can launch products.
Nice. Andre comments, this is a complicated process. You need to have a polymer and a UV light. And then Michael asks, can they be mixed with paint and still work?
We think paint's an ideal way of using these, because as paint dries, materials like our polymers would tend to go to the surface and be in a surface layer. So even though you have a layer of paint that's fairly thick, we'll get the most bang for our buck by the fact that it actually migrates in the drying process towards the surface. So it's really ideal for employment in the paint.
And as we said, you don't need UV light, right? We can do this in room light or sunlight. Some of the compounds are still activated in fluorescent or other kinds of white light.
Can I say thank you to everyone? That was very impressive. And I do like the collaboration between the medical school and the School of Engineering. So thank you to the speakers, but I wanted to mention a couple of things. Okay, there is a question. I will allow you to answer, but first I want to say that John Farris is one of the people who's listening, and he happens to be the son of the dean that we named the building Farris, the Farris building for. So it's nice to have you, John. And I will still let you answer, but I don't want to forget to say, Mariah, thank you for hosting and taking care of this series this semester. We've never done it before, but unfortunately for us, good for Mariah, that's her last semester. She's graduating, so congratulations. So now I'm gonna let you answer. Go, Mariah. Sorry to interrupt, but I didn't want to forget that, so.
Cool, thank you. So we have a question from Fernando. He asks, did you all take into consideration the condition of solar urticaria, a type of UV light allergy that, although rare, can cause negative effects on some individuals?
We weren't really developing this for human application, right? Our idea was materials and not necessarily putting it on our bodies. Honestly, no, we didn't think about that.
But it's good to know.
It's good to think about, right?
Okay. If there are no further questions, I don't know if we want to get, anyone else wants to get in some last minute questions. Okay, we have one from Karen. What is the effect of temperature on them? Like if it's used in an unexpected heated room?
Temperature effect is probably very small. The excited state processes such as we're using are very little affected by temperature.
And also, as materials, these are non-volatile. So this is what gives them the long lasting effect. And also these are fairly stable because these are polymers and oligomers. So comparing to some other compounds that are in development, these are pretty robust and stable compounds. So in terms of in a heated room, 10, 15 degrees, it's not going to make that big of a difference.
Mm-hmm.
So I did put a link in the chat. That's for an event that's happening tomorrow. The ESS is holding that. So any of you who are interested in that, you can click on that, or you can just visit, type in UNM ESS events. You can find that for tomorrow. It has a Zoom link, it has a flyer, the information for that. But thank you all for attending today's Engineering in Action talk. Our next talk will be on June 25th at 3 p.m. And this one will be focusing on our Center for Water and the Environment. We look forward to seeing you all again. Thank you again for joining us today. Stay safe and go Lobos.